During interphase, DNA replication occurs in readiness for meiosis. The DNA forms chromosomes with a homologous pair of chromosomes, being one maternal and one paternal chromosome. In prophase 1, the centrioles create poles with spindle fibers slayed between them. The homologous chromosomes pair up to form bivalents, with the place where they connect being the chiasma. The centrioles move to opposite sides of the cell. At this point, the nucleolus begins to fade. The chromatids stationed at the chiasma break and then join with the homologous chromatids so the genetic information is swapped between them. Metaphase 1. The chromosomes line up along the equator of the cell and their centromeres attach to the spindle fibers. Anaphase 1. The spindle fibers contract towards the poles, pulling the whole chromosome apart. The chromosomes reach the poles of the cell. Each pole now has half the number of chromosomes present in the original cell. The cell membrane constricts, dividing the single cell into two daughter cells. Once again, the centrioles create poles on which the spindle fibers form. The centrioles move to the opposite sides of the cell. The chromosomes line up on the equator with their centromeres attached to the spindle fibers. The spindle fibers contract towards the poles. The centromeres split with the individual chromatids being pulled to the opposite sides. A new nucleus forms around each pole. The cell membrane constricts, dividing the single cell. By the end, 4 haploid daughter cells have been formed, with each daughter cell having half the number of chromosomes of the original cell. Importantly, all the daughter cells have different genetic setups to each other and to the original cell.